Hello. Can we come? Can everybody come and sit, please? We are going to start the session. My name is Michel Maréchal, and I will chair this session. I have attended previous CECAM celebrations. I have even organized, or I would say disorganized, CECAM50. And I remember having presented a talk at CECAM50. And that talk was about the application of molecular dynamics to hydrodynamics, to the simulation of hydrodynamic instabilities. Now, having organized CECAM50, I have to say that the quality of CECAM celebration is going up tremendously. And I wish to congratulate all the team, Ignacio and all the team that has organized this one. And having presented a paper on hydrodynamic instability, I'm very happy to chair the session where Julia Yeomans will speak about it, because she has become one of the leaders of this field of mesoscale, I would say mesoscale hydrodynamics and the applications to active matter, to hydrophobicity, and everything which is important on the mesoscale. So she is professor at the University of Oxford, and I wish to point out that she was saved from the Ising model and Michael Fisher that was her field at the beginning of the career. So Julia, please, you will tell us what is
very
topology.
Much, yeah. Yes.
In
and I used to hate biophysics as well, and look where I've ended up. Yeah. When I was trying to write this talk, I realized that there are a lot of different numerical methods in here, and we wouldn't have been able to use them at all 50 years ago. And the fact that we can use them now to sort out the sort of physics I'm going to talk about is due to people like you at places like CECAM. So happy birthday, CECAM, and thanks to all the people who have been sort of behind the sort of things we can do today. I'm going to talk about active matter. What active matter is a system which takes energy from its surroundings, and it does this on an individual particle level, and then uses it to do work. And so it's anything biological, really. And in particular, it's too hard for us to look at people or cows or robins and things, so we have to look at small biological things, for example, cells, molecular motors, or bacteria. So I'm going to start by just trying to explain why I think it's interesting to look at active matter. I mean, cells are really complicated things, and we're essentially engines, and it's the cells, what's going on in the cells inside us, which is making us being able to breathe, to eat, to move, to do everything. And in particular, we need to worry about little motors which move the cellular components around, which move around all the different proteins that are needed in the cell. And here we have it. Here's the molecular motor. That little thing there, with its little feet there, is a molecular motor. It's walking along a track which is being laid down in real time. It really is like this in cells. It's not just a fancy movie. It has to move along tracks because it's tiny. This thing is about sort of 15, 20 angstroms in size. And so it would just get buffeted around by the Brownian motion if those tracks weren't there. So somehow the cell is managing to lay down these tracks, to have these little motors walk along, and then to work out which proteins the motors have to move around. People understand pretty well how these things walk. These are proteins. They have a certain structure. They gain a molecule. The back foot gains a molecule of ATP. That means it can detach. Because of the shape of the protein, it tends to move forwards, and then it can grab the microtubule ahead of it. I think nobody understands the control theory behind this. How does the cell decide how it's going to sort everything out? And so there's some really, really big, complicated questions there. And they're very important questions, because if we knew how things like this work, we could use them as tiny molecular motors, and we could also probably do all sorts of exciting medical things to help cure diseases. I'm afraid that's too hard at the moment, so let me tell you first about a synthetic system based on these microtubules, these tracks, and the molecular motors, which was made about 5 years ago now in Zvonimir Dodžik's group. So what Professor Dodžik did is take these microtubules, these green things are the microtubules, the tracks, and put them in a solution with a large number of molecular motors. The motors were special because they had two heads. They bridged between the tracks. And these motors can move just in one direction along the tracks. So if you have two microtubules next to each other, with some of the motors moving that way on this track, and motors moving this way on this microtubule, what's going to happen is that the microtubules are going to get pushed around relative to each other. You're going to have an active system because you're putting in ATP to drive them, and then that's going to be translated into motion by these molecular motors. If this system is dense, what happens is this. What you can see, these little sort of stringy things, are bundles of microtubules, and they're being moved around by the molecular motors, and this is a state which we've called active turbulence. It sort of makes sense that something has to happen because you're pumping in energy, but it's not so obvious what. If you look also at a slightly different system, larger now in length scales, these guys are bacteria, so micron length tiny swimmers. This is a layer of these swimmers. Again, they're active, they're swimming around, and you get this this state which looked a bit like turbulence, which looks like active turbulence. And it would be nice to understand if there was any similarity between those two states and how to explain the sort of thing, the sort of states that we end up with. Okay, so that's why study active matter. Okay, and probably I'm going to really concentrate on the last one here. These things are out of equilibrium. Living systems are out of equilibrium. If they stop being out of equilibrium, this is really bad for them. And so things like the collections of microtubules and motors, things like the microswimmers, are examples where you have a large number of particles in a system which is out of equilibrium. And so they're a very nice example of non-equilibrium statistical physics. So what am I actually going to do? Well, it's always a bit difficult talking to a large and mixed audience, so hopefully there'll be something here for the experts and the non-experts. I'm going to start with some background and try to explain to you where that active turbulent state comes from, and in particular to focus on the fact that it's related to topological defects, and in particular motile topological defects, ones that move. I'll then talk about some recent work which is pretty much finished, which looks at these defects in carpets of bacteria. This is the story of the hare and the tortoise. And then for the experts at the end, I'll talk a little bit about some work we did a little while ago, which we're carrying on with now, where I really don't understand what's going on. So there are more questions than answers in the last bit. So let me start with some background. Pneumatic symmetry. This is pneumatic symmetry. A thing like that is pneumatic because it has a preferred axis, but not a particular direction up and down. And if you put a lot of pneumatic molecules together, you end up with the possibility of a pneumatic phase. Things tend to align parallel to each other. This is what you very typically see in liquid crystals and laptop screens. They align parallel to each other because the entropy then, entropically this is favored because then they have more room. And the order parameter Q is a tensor which will describe the state of that pneumatic order. Its largest eigenvalue tells me how ordered the things are, and its largest eigenvector tells me the direction of the order. We know about this at Oxford because in the summer when it's warm, you end up with the isotropic phase, And then in the winter, when it's cold, you end up with a pneumatic phase. So there are two really nice things about these liquid crystals. The first is that they are an elastic liquid. If you try and muck up the pneumatic order in some way, for example, here you can see that we're trying to bend the pneumatic order, then there's an energy associated with that, and they're going to try and pull those plates back so they're aligned properly as a pneumatic. There's an elastic energy, and you can write down something which looks like a derivative of the order parameter squared as a free energy which describes that elastic energy. The other nice thing about them, and this is a really important one, okay, so this is going to be important for later on, is that you can get topological defects in these systems. Topological defects are defects in the pattern of the pneumatic order. The pneumatic is not all nicely lined up. You get these special points. They're called topological because they muck up the ordering all the way to infinity, and so they have an infinite energy associated with them. And in a perfect pneumatic, you can't therefore just have one of them. They have to come in pairs, and in this case would be a plus a half and a minus a half pair. Can you remember the shape of these things? Plus a half is this thing which looks a bit like a comet. This minus a half has a threefold symmetry. If you have a plus and minus a half defect in a normal passive liquid crystal, what happens is that they attract each other with a logarithmic attraction, and they will eventually annihilate, and you get rid of them, and you get a nice perfect crystal, and then you're happy because your laptop screen isn't mucked up. The other thing we're going to have to know about is the equations of motion for this liquid crystal hydrodynamics. So what I've done here is write down a sketch of the continuum equations that you need to describe the hydrodynamics of liquid crystals. So it's like Navier-Stokes, only for liquid crystals. I'm not going to worry too much about details, but this one here is the equation of motion for the order parameter Q. These pneumatics are moved with the flow, they're turned with shear flow, and then they can relax to the minimum of some sort of free energy. And then you have the Navier-Stokes equations here, which is the Navier-Stokes-like equation for the velocity, usual inertial term, and on this side, the gradient of the stress tensor, and this is the stress which is related to derivatives of the velocity. Now, you might well be asking, why is she batting on about liquid crystals when she's meant to be talking about active systems? Okay, I think at this point, that's a very fair question. So let's get back to the active systems. And there we are. This is what we're trying to explain about. We're trying to understand what's going on here with this active turbulence. Well, these swimmers are pneumatic in shape, right? They're long and thin and pneumatic. So it sort of makes sense that if I want to write down the continuum equations of motion, what I can do is write down the equations using these pneumatic continuum equations, which are well understood. But you have to have something else in there as well, because the swimmers, these little swimmers, are producing flow fields. So each of them is going to feel lots of flow fields from all the other swimmers, and that's going to give you an extra stress, extra forces, extra stress in the equations of motion. And it turns out that the flow fields produced by these swimmers has pneumatic symmetry. So here's a little swimmer. These are some beautiful experiments from Ray Goldskin's group, and you can see that the swimmer pumps fluids out from its ends and pulls it in from the sides. I've got a preferred axis, but no preferred forward and backwards direction, at least on long length scales. And so what I've got here is something which is pneumatic. And so it makes sort of sense that if you want the extra stress produced by that thing, it's going to be proportional to the pneumatic order parameter tensor Q. Makes sense. I haven't proved it. You can prove it, okay? If you want to add in the effect of all these little swimmers, what happens is that they give you an extra stress. The extra stress looks like this, some coefficient which tells you how many of the things there are, and then a Q which takes account of the shape of the extra flow field that the swimmers are producing. So if I want to write down the continuum equations for one of these active systems for an active pneumatic, it's just the same as a passive pneumatic except for this one extra term, and where that one extra term goes is in here, in the stress. And that stress comes under a derivative. And so what that means is that any time the pneumatic order is screwed up, any time the pneumatic isn't nice and regular, any gradients in the magnitude or the direction of the pneumatic order, you're going to get an extra force and you're going to get flows. Gradients in the magnitude or the direction of the pneumatic order parameter induce a flow. This is how it works. You start off with a nice pneumatic. There's some sort of fluctuation. Hello, why aren't you working? Probably because I'm pressing the wrong thing. No? Yeah, there's some sort of fluctuation. Whoops, pressing the wrong thing. Some sort of fluctuation. That induces a flow because I've got a gradient in the pneumatic order. The flow happens to be this way, and so things get worse and the whole thing goes wobbly. Essentially, that's saying if we do a linear stability analysis, the pneumatic state is unstable. And so I've got to get something else. And what happens instead is this active turbulence. And then you're in a bit of a mess because you can't do the maths anymore. So you have to take those equations. And because we're from CECAM, we know how to solve them numerically. And if you do that, what you end up with is a picture like this vorticity field here. It shows that that state has a large positive vorticity in red, large negative vorticity in blue. That's places where you get the flow field swirling a lot. And the length scale of those vortices is about 5 to 10 times the scale of the microswimmers. You're getting collective behavior on a large scale compared to the individual particles. And if I look a bit closer at what's going on, what you can see here is that I have a bend in stability, a bend here in the director field. The director field is bent, and that's what's giving us stresses, which is giving us the flows, which then swirl around and give you active turbulence. Okay, so this is a liquid crystal, right? It's meant to have topological defects. Let's look at the topological defects in that simulation. The red ones are the pluser halves, and the blue ones are the minuser halves. And what they're meant to do in a passive pneumatic is come together and destroy each other. And if you look carefully, that's what was happening there. But if you look, if they're all destroying each other, there should be less of them. And that's not happening. What's happening is that they're the same number at the end as at the beginning. And let's see if I can find it. Up here, something else is happening. They're also popping out like that. The topological defects are not just destroying each other in pairs, they're also appearing in pairs. That's fine, because you're pumping in energy, so that's allowed. But then, if you think you'd make a couple, what's going to happen is they will immediately destroy each other. And the reason that doesn't happen is that the pluser half defects in particular are self-motile. They're created, and then they move away. And we understand why that happens, because here, there are lots of gradients in the order parameter. It's changing its direction. And so there's stresses, and so there's flows, and that propels the defect. This one here, there's stresses, but because of the symmetry, they tend to balance each other out. And here's a picture of the flow field around those defects. Sorry, I can see it on here and not there. Here's a picture of the flow field. The minuser half one, you get a very obvious 6-fold symmetry. And the pluser half one, you just get this thing zooming off on its own. So now I can look back at these very nice experiments from the Dogic group, and you can see, I know it's easier when you're a bit more used to it, that what's happening is topological defects are forming and moving away from each other. Here's a still to make it a bit easier. As these microtubules bend, it's like having a minus defect here and a plus one there, and they move away from each other. And so this active turbulence state is a state of defects continually being created, moving around, and being destroyed. Right. So then I'm now going to tell you two stories about how it seems like these defects actually exist in real biological systems. And maybe they're important biologically. The jury's still out on that one. This work has been done by some splendid collaborators. In particular, Armin's just moved to the Niels Bohr Institute, and he did all the hard work on the simulations and the theory, and Ollie has just got his PhD, and despite the photo, is an absolutely superb experimentalist. Okay, so this is what we're looking at. These are Pseudomonas. They're little bacteria. They're probably about 2 microns long. They don't actually swim. They move by twitching, which means they have tiny feelers which they use to pull each other over the surface. You can see that again you're getting a state where you have short-range pneumatic order. These things are pneumatic shaped. And you're getting something which looks pretty much like active turbulence. The question that our zoologist colleagues were asking was, if you have two different sorts of bacteria, how does the colony grow? How does the colony of bacteria spread? The reason they're interested in this is that it's a big deal at the moment in zoology to look at systems where you have more than one sort of bacterium. They're sort of working up to what's happening in our tummies, where there are about 100 sorts, but we're starting with 2. So the 2 different sorts are the normal wild type, and then ones which have extra pili, and so they can move faster. So the 2 different sorts are basically a slow bacterium, which is the normal wild-type bacterium, and then this hyperpileated one, which moves about 2 times faster. And if you look at the probability distribution of the speed, these ones here are the faster ones with more pili. So here's a race between the 2 different sorts of bacteria. At the top are the wild type, which is slow. In the middle are the faster ones. And what's happening is that there's a sort of lots of them behind here, and so they're expanding because they're moving. And most of the expansion is due to motion rather than cell division. Okay, so something's going strange there, right? The slow ones are winning. Hare and tortoise story. You need to think about the hare and the tortoise for this. For some reason, the ones at the top, which are the slow bacteria, overtake the fast bacteria and spread faster. Those lines are just where you move the microscope stage, and they didn't cheat, I hope. Okay. This just looks at the front of the colony, and as a function of time, you go from having fast ones at the front, which are marked yellow, to having the blue ones, the slow ones at the front. So something is stopping those fast bacteria from winning the race to colonize the surface. Okay, so we're talking about topological defects. Let's just stand back a minute and look at topological defects in this system. It's a pneumatic. You can identify topological defects. These are topological defects in the experimental system. The red ones are +0.5 and the blue ones are -0.5. We can model those. This is a very simple model, just hard rods each rod is subject to a constant driving force, and then there's an excluded volume interaction between them, and they look pretty much the same, and you can identify plus defects and you can identify minus defects. This is just the picture I showed you before. This might be another way of modeling what's going on. This is a continuum version of what's going on. And I think it's a very fair question to say, so what? Are the defects in these three different cases, are they the same thing? Are we looking at the same sort of thing here? So what we did is we measured the flow field around the topological defects in the continuum models, which I've shown you before, in this rod model, and in the experiments. For the -1/2 ones, here's the nice six-fold symmetry of the flow field around a -1/2 defect. In the rods, you see the six-fold symmetry, not quite so nice because you have noise in these simulations. And in the experiments, again, the six-fold symmetry. Similarly, the +1/2 ones. Here, in the continuum simulations, looks pretty much the same. In the rod simulations, looks pretty much the same in the experiment. So it seems sensible that at least at this level, we're seeing the same active topological defects in the experiments, in the rod model, and in the continuum simulations. So what's this got to do with what's happening in the layers, the hare and the tortoise story? Well, this is a picture of those fast bacteria as a function of time, and then it starts again at the beginning here and carries on. And you see blobs forming. What those blobs are places where the bacteria are pointing up from the surface. So instead of moving on the surface, they nucleate regions where they're pointing upwards, and then those regions grow and they get stuck. Basically, they get stuck in these standing up regions, and when they get stuck in the standing up regions, that means they're taken out of the pool of bacteria that are allowed to expand. This happens for the fast cells, but it doesn't happen for the slow cells. So why do the fast cells nucleate standing up regions? And the answer is topological defects. What happens is that two +1/2 defects come towards each other. They repel each other, but they're moving, so they can get pretty close. They form a virtual +1 defect, and that gives the system the energy to stand up. Here it is in the simulations. If these things are moving towards each other slowly, you get what happens on the left. They stand up, but then they fall back down again. If they're moving fast, they stand up. These blue ones are pointing towards you, and then they stay stuck, pointing in that direction. Bigger simulations. As things move faster, moving from left to right, you get regions where these things are, well, you probably say verticalized in the States, but it's not a word. Okay, and you end up with them all pointing up like that, and then very few are left to spread out. I showed you the simulations first because they're a bit easier, I think, to see what's going on. Ollie managed to find one of these regions in an experiment, so these are experiments now, but it's sort of quite difficult to see, and you can imagine how difficult it is to find as well. This thing on the left, let's see if the movie will move, you can just about see forming in the middle this yellow region here, which is the verticalized region. And if you're a zoologist used to looking at this thing, it's pretty obvious to see what's going on. And so what we've found here is that we get this strange hare and the tortoise story. Bacteria which are individually slower can expand faster when they're in colonies. And that's because you get these virtual +1 defects which act as nucleation sites for the bacteria to form these immotile clusters. And if you're a zoologist, you immediately say, right, bacteria have evolved to move with a certain speed, which are the speed of our slow ones, because those are most efficient, but they haven't evolved to move faster, which you might think was better, because you have this problem that they then get stuck. And maybe, well, I think this is probably one of the ways that biofilms can form. So this is a picture to remind you of what's going on there. Right, I wonder how many times they had to run that race till they got the right result. Right, so that's the second story. Now the third story is going to be, the first bit of it is published, and then it led to some questions, and so the second bit I'm going to talk about to a pretty good approximation. I don't know what I'm talking about, but I think there's some really interesting questions. So now we're looking at eukaryotic cell layers. Eukaryotic cell layers are the squishy cells which cover the inside of most of the organs of the body. And again, if you look at two-dimensional layers of these plated on a Petri dish, you get something which looks pretty much like active turbulence. So we thought that it would be a good idea perhaps to look at topological defects in this system. And the we is these people, and in particular, Thuan Beng So did the experiments, and Armin was responsible for the early theory, and then later Roman and Guanming. So, I mean, the first problem is that these cells are circular. So how am I going to talk about them in terms of a pneumatic? Well, the answer is they might be instantaneously circular or circular on average, but they are pulled by each other because there's cell junctions between the cells, so the cells pull themselves around, and so instantaneously they have a pneumatic shape, and you can associate a pneumatic order parameter with them. And if you do that, you can indeed identify topological defects. Here's a +1/2 defect, and it's a motile +1/2 defect. We measured the velocity field around these cells, looked the same as the ones I've shown you before. We also measured the pressure field around the cells. And what this slide shows is that if you look at the pressure field and you compare the experiments and the simulations, what you end up with is pictures which look very much the same as each other. So they really do look like active topological defects. You can also do this nice experiment, which shows that these defects are active. The graph I've got here is the number of defects as a function of time. Number of defects as a function of time. And here, the activity is switched off by adding blebistatin, which puts the cells to sleep. And the number of defects goes down. Here you wake the poor little things up by washing out the blebistatin, and the number of defects goes up again. So these things are behaving like you'd expect active defects to behave. And we actually found a correlation. There may be a biological relevance here. We found a correlation between the positions of the +1/2 defects and places where cells died and were pushed out of the layer. The rationale for this correlation is that at the topological defects, you get an extra stress. This stress moves a chemical, which has the wonderful name of YAP, from the nucleus to the cytoplasm, and that's a signal for cell death, and then the cells are pushed out of the layers. But, you know, why these circular cells? I mean, why are they pneumatic? This isn't right? You've got circular cells, and we're wittering on about how we're seeing this active turbulence, that we've seen in pneumatic systems. So we wanted a numerical approach which would help us to understand what was going on. And so what we're trying to do now is develop a phase field model of these cells. And let me just show you, it's very slow, but it gets there in the end. This thing moves, okay? You can see the cells slowly moving around. And because we can look at individual cells, that will help us to understand on a much more detailed level what's happening in these systems. We built on work by Martin Grant and Igor Aronson, and let me just spend a couple of slides telling you what this phase field does and how it works. Each cell has a field phi I, a phase field associated with it, which is 1 inside the cell and 0 outside the cell. This phase field has a dynamics. It moves with a velocity v, and it relaxes to a minimum of a free energy. And because this is an overdamped system, whoops, wrong way, because it's an overdamped system, what happens is that the velocity is just proportional to some external force. The free energy is very like the sort of free energy you'd have if you had a set of bubbles. First of all, we have a term which defines the interface around the cell between the inside of the cell and the outside of the cell. Then we have a soft constraint on the area. Physically, that makes sense. Then we have a repulsive interaction to stop the cells overlapping with each other. And sometimes we also have an attractive interaction which says that the cells like to be stuck to each other. And from that free energy, I can define a passive force density. If I just ran that simulation, I'd have a load of bubbles. This is an active system, so what I need to put on is some sort of active force. And remember that the active force had the symmetry of the Q tensor Q, and it's sort of easy to do that. I'll just show you the maths on this slide, and then on the next slide the picture, which is easier to understand. I can very easily work out the deformation tensor of those cells. It's related to the gradient of the phase field. That makes sense. The gradient of the phase field is non-zero around the edge of the cell, and so from that you can work out the deformation, and that deformation is essentially the same thing as this Q tensor. And so I can put on a force which is proportional to the Q tensor. This force here is essentially the same thing as in the continuum models. But the easiest way to see it is that it's a force acting on a cell due to its neighbors, which has this pneumatic symmetry. It works in the direction of the extension, and it either pulls outwards in the direction of the extension and inwards from the sides. That's called an tensile force. Or it can go inwards at the ends and outwards at the sides, and that's called a contractile force. And what happens is if this system is extensile, you get active turbulence. And the reason is that a tiny fluctuation is going to be enhanced by this force. Okay? If it starts being a tiny bit perturbed, then what happens is the force is going to pull it into a pneumatic shape, and you can get active turbulence. If you have one of those contractile systems, you've had it, because any tiny fluctuation will just get pushed back and it will become circular again. And in fact, we looked at our cells and we found that the defects were extensile-like defects, both in the experiments and the simulations. This just shows you the active turbulence in these systems, which is meant to move. You can see the topological defects, you can see the vorticity fields, and you can also see, again, if you look around the cells, the flow fields around the topological defects, which have the right sort of symmetry. So we understand why in a cell layer you can get active turbulence. It's because of this bootstrap effect. The cells exert forces on each other, which once you've got a tiny distortion, pull them out into having a pneumatic shape. But that's not the whole story. That can't be the whole story. Because if you have a single cell, right, it moves. It doesn't just change its shape because something's pulling it. It actually moves. And this is an excuse to show you this beautiful movie where they're trying to show you how well they can do the graphics, and quite rightly. Okay, here it is moving through the extracellular matrix in the body. If this thing's moving, it has to have a net force on it. And so we have this strange question that if you have a single cell, it has a net force on it. The equal and opposite forces because it's pushing on the surface. Whereas if you have a cell layer, all the forces are balanced. The cells exert on each other. So let me finish by showing you some beautiful experiments, which I think we don't really understand, but they're very beautiful, which was from the group in Milan. They looked at a cell layer. The cell layer is doing active turbulence like we've seen cell layers do. They then added a chemical, this RAB5A, which is meant to muck up cell junctions, and the cells just took off. A flocking type thing. The velocity is enormous and they all move. Let's just compare those two situations. That's the one where the cell junctions are behaving like they ought to. This is one where somehow the cell junctions are mucked up. And so we have to understand the relation between, basically we have to understand what's going on here. We have to take our phase field model and add in forces, and how to add in the forces is what we're trying to understand at the moment. So let me finish with something which is for CECAM for the next 50 years. How do we model this? This is Drosophila. It's a fruit fly, and it's growing up to be a grown-up fruit fly. Those sort of dots you see are nuclei. We've got cell division. We've got cell motion of cells which are connected, very like the ones I showed you. We've got hydrodynamics. This thing looks like it's flowing, and presumably there's some sort of chemical signaling. This is another of the places where nature is much better at doing things than we are, and understanding how she does it would be it's interesting to try and find out. Thank you very much for listening.
Thank you very much, Julia. Much of the simulation that you have shown are two-dimensional.
Yeah.
Is this also true for the experiments, or is the third dimension of any relevance?
The experiments up to this year have all been two-dimensional. You tend to put this stuff on a surface. And even with the swimmers, people want to do 2D because then you can image it much more directly. But just actually last week, there was a paper on the archive, again from the Dogic group, where they've been looking at 3 dimensions. So up to now, yes, but people are starting to look at 3D experimentally.
The paper is open for discussion, so questions from the audience?
In the case of this, here, the bacteria,
Sorry.
the second case that you have...
Where am I looking? Yeah.
Here.
Sorry.
For the fast bacteria, you have these one defects that are stable. I was wondering how they can be destabilized, whether interacting with other half defects. And in the continuum model, it's not obvious to see this one defect. So I don't know when you can see that in the continuum model.
The +1.
Yeah, the +1.
Remember that, I mean, certainly if you just have free energies, 2 plus a half, which will come to each other, if they're allowed to point up, will point up just on free energy grounds. Don't actually think that's what's happening here. What happens is they're coming towards each other, and they will flick up. It's really if you have a continuum model which allows it. So if you have an order parameter which is allowed to go at 3D order parameter, whereas many of the continuum models have a 2D order parameter. A +1 defect in 2D will look pretty much like a star or like a loop like this. But it never gets that far. It always just pops up before it ends up that shape.
Ralph?
It's not so easy to swim at low Reynolds numbers. Is that why your swimming bacteria in the first part were pneumatic and not polar?
I mean, swimming at low Reynolds number, you have to obey the scallop theorem, which says that the swimming stroke is non-invariant in time, which is a little bit different from the story here, because I was talking about far flow fields. If they're polar, the far flow field will be the same. So the hydrodynamic terms you need in the equations of motion are the same. The current argument is that if you average over a large enough area in the continuum, a pneumatic theory is okay. But there are questions, I think, still about pneumatic versus polar. I don't completely understand it, and I'm not sure anybody does.
Further question?
From there?
There is.
Oh, no.
I was wondering whether active turbulence how active turbulence is compared as normal turbulence. So if you have any insights on the turbulence spectrum or something like that.
Yes, we and many people have measured that, and I think to a pretty good approximation, it's not similar. You don't get the same sort of scaling as you'd expect, because here you're shoving in the energy at small scales, and active turbulence you're putting it in large scales. And I'm afraid calling it that has given me a lot of grief. I mean, I meant turbulence as in chaotic sort of behavior, but the inertial turbulence people are not happy because they mean something, they mean real inertial turbulence. Then you call it chaos and you've got problems with the chaos people, so I'm not sure what's right.
Well, you don't have so many length scales, so you cannot compare really to the fully developed turbulence or any scaling theory for turbulence.
That's right. The energy comes out on the vortex length scale,
yes,
and so you just go from small to vortex. So you don't see proper scaling at all.
Do you see any need of a particle simulation? That, I mean, all what you have explained so far, I think, is really continuum modeling. Do you see anything which would be a particle simulation?
I mean, the things we did for the bacteria, where we just had rods, is an agent-based particle simulation.
You mean
and
the
lots of other
Yukawa
people.
potential? Some Yukawa potential, you call it, or something?
Yeah,
Yeah.
just rods, which are hard rods, yeah. And there has been a lot of people who've done particle-based simulations. And it's all adding up to our understanding of these things.
There is a question there.
Really amazing talk. Completely new on this topic, but where you were showing this annihilation of this type of defect, have you studied or do you know if there is some, for example, when two of these defects they annihilate, I will imagine that they release some kind of energy, or they have some energy associated when they are created. Yeah. So, I mean, can you comment a bit on this?
Yeah, I mean, that's quite true. I mean, the reason they annihilate in a passive system is that they then are minimizing the energy. And the reason they're created is that there's enough energy to do that, because in an active system, you're pumping in energy. So yes, that's certainly the case. I mean, I haven't talked about really a theory of these defects. I've told you what happens, but we still need a theory of these active defects, which we don't have.
So do you think there is a way to monitor, for example, as in the case of, for example, in Grand Canonical, you can monitor the populations of every of these different looking at these things like particles, and but also at the distribution of the energies associated with these like virtual particles, if you want to look at that. So
yeah, I mean, people are trying to write down a particle version of this active turbulence, thinking of it as a gas of topological defects. The reason it's hard is that you're not in thermodynamic equilibrium. So all the normal stories go out the window.
all right, thank you.
Yes, there.
Hi. I didn't understand the experiments with Pseudomonas. Did you perform the experiments, or?
No, it was Ollie Meakin
In Oxford.
in Oxford
Yeah.
in the zoology department.
So
yes.
is this Pseudomonas which have pili on both ends, or all along the body, or?
They have pili all the way round, and then the ones with more feet are genetically modified to have more pili. Yeah, Kevin Foster's group in Oxford who do a lot of these
and
things.
because usually Pseudomonas, when they crawl, they do stand up, but this is prior to detachment, and then they swim away. Did you see in this experience, was there something like that? These clusters detach, or never? Or
no, and they wouldn't, because these are between two bits of gel, which you need to feed them.
Okay.
Yeah.
Thank you.
If there are no further questions, I think we can close by thanking Julia very much for this very nice talk and all the speakers of the afternoon.